Genetic Disorders by Adolfo and Elena. Sex-linked genes exhibit unique patterns of inheritance. There are two types of chromosomes, X and Y. Female egg cells all contain X cells. However, male sperm cells split into X and Y. Half of the sperm contains the X chromosomes and half contain the Y chromosomes. If the egg cell is fertilized by an X-containing sperm cell, the zygote will be a female. If it is fertilized by a Y-containing sperm cell, the zygote will be a male. So basically, the sex of the embryo will depend solely on the sperm cell. Inheritance of X-linked genes. Fathers pass X-linked alleles to all their daughters but none of their sons, while mothers can pass X-linked alleles to both sons and daughters. However, even though it is rare for a female to have sex-linked disorders, it is still possible. If the father is colorblind and the mother is a carrier for both recessive allele, then a female can be born with sex-linked disorders such as colorblindness. This happens because males have only X chromosomes, so being homozygous and heterozygous does not exist in this case. Sex-linked disorders. An example of a more serious linked disorder is Duchenne muscular dystrophy. This disease is characterized by progressive weakening of muscles and loss of coordination. Another example is hemophilia. When a person with hemophilia is injured, the bleeding will be prolonged because a firm clot is slow to form. This is caused by absence of key muscle protein called the dystrophin, and the gene of the protein is the X chromosome. Linked genes tend to be inherited together because they are located near each other on the same chromosome. Linked genes are genes located near each other and tend to be inherited together in genetic crosses, as they are genetically linked. To distinguish between sex-linked genes and linked genes, remember that a sex-linked gene refers to a single gene of a sex chromosome, while linked genes refers to a two or more genes on the same chromosome that tend to be inherited together. Morgan's Drosophila experiment. To see how linkage between genes affect the inheritance of two different characters, let's examine Morgan's Drosophila experiment. Morgan wanted to know whether the genes for body color and wing size are genetically linked, and if so, how this affects their inheritance. The experiment consisted of gathering a wild-type gray body with normal wings and a double mutant with black body and vestigial wings. The result was a F1 dihybrid test cross with a wild-type F dihybrid with gray body and normal wings, then mated with a homozygous recessive black body and vestigial wings. The results were a wild-type gray normal, a black vestigial, a gray vestigial, and a black normal. Morgan's Drosophila experiment conclusion. Since most of the offsprings have had a parental P generation phenotype, Morgan concluded that the genes for body color and wing size are genetically linked on the same chromosome. However, the production of relatively small number of offsprings with non-parental phenotypes indicated that some mechanism occasionally breaks the linkage between specific alleles of genes on the same chromosome. Genetic recombination and linkage. Genetic recombination is the production of offsprings with a combination of traits that differ from those from either the P generation parents. Recombination of unlinked genes, independent assortment of chromosomes. Mendel learned from crosses in which he followed two characters that some of the offsprings have combinations of traits that do not match those of either parent. Half of the offsprings in the Punnett square are parental types, offsprings who are expected to inherit a phenotype that matches either of the parental phenotypes. The other half are recombinant types of offsprings who have new combinations of phenotypes. Genes crossing over. Crossing over is a process in which the physical connection between specific genes on the same chromosome is broken. This process accounts for the recombination of linked genes. New combination of alleles variation for natural selection. The abundance of genetic variation provided by recombination is the base of which natural selection works. If the traits conferred by a particular combination of alleles are better suited for a given environment, organisms possessing those genotypes will be expected to thrive and leave more offsprings, ensuring the continuance of their genetic complement. Genetic maps. A genetic map is an ordered list of genetic location of the gene, along a particular chromosome. The further apart two genes are, the higher the probability that a crossover will occur between them, and therefore the higher the recombination frequency. A linkage map is a genetic map based on recombination of frequencies. Alterations of chromosome number or structure cause some genetic disorders. Abnormal chromosome number. Non-disjunction is an occasional mishap in which the member of a pair of homologous chromosomes do not move apart properly during meiosis I, or sister chromatids fail to separate during meiosis II. When this happens, one gamete receives two of the same type of chromosome, and another gamete receives no copy. If either the aberrant gametes unite with a normal one at fertilization, the zygote will have an abnormal number of particular chromosomes, a condition known as aneuploidy. Aneuploidy may involve more than one chromosome. Abnormal chromosome number. Fertilization involving a gamete that has no copy of a particular chromosome will lead to a missing chromosome in the zygote, and the gamete will have only one chromosome, and the cell is said to be monosomic. Fertilization is involving a gamete that has two chromosomes already and unites to have three chromosomes, is said to be trisomic. People who have three copies of the chromosome in chromosome number 21 are born with Down syndrome. Alterations of chromosome structures. Errors in meiosis or damaging agents such as radiation can cause breakage of chromosome, which can lead to four types of changes in chromosome structure: deletion, duplication, inversion, and translocation. Deletion occurs when a chromosomal fragment is lost. Duplication, when a deleted fragment becomes attached to an extra segment to a sister chromatid. These first two steps are especially likely to happen during meiosis. Inversion, when a chromosomal fragment reattaches to original chromosome but in reverse orientation. Translocation, when a fragment joins a non-homologous chromosome. Human disorders through chromosomal alteration. Down syndrome is the result of an extra chromosome 21, so that each body cell has 47 chromosomes. Klinefelter syndrome is when there is an extra X chromosome in male producing XXY, People with this disorder have male sex organs, but the testes are abnormally small and they are sterile. There are also men who are born with XYY, but these undergo normal development. Women can be born with XXX and with only one X. Women with trisomy X are slightly taller than normal and are more propense to learning disabilities. Women with X are also sterile. Even though the extra X is inactive, some breast enlargement and other female body characteristics are common. If you look closely into chromosome number 21, you will notice it has 3 copies of chromosome instead of 2. This causes heart defects, development delays, characteristic facial features, and increased chance of developing leukemia and Alzheimer's disease. Disorders caused by structural altered chromosomes. Deletions in human chromosomes cause severe problems. For example, Cri-du-Chat syndrome resulting from a specific deletion in chromosome 5. This disease occurs when reciprocal translocations happen during mitosis of cells that will become white blood cells. Children with this syndrome are severely intellectually disabled and have a small head with unusual facial features. Chromosomal translocation have been implicated in certain cancers, for example, chronic melangineous leukemia.